Hi, and welcome to a learning module on fermentation. So, fermentation is the process of metabolically converting a carbohydrate such as starch or sugar into an alcohol or acid through the use of microorganisms. There are many examples of fermentation that occur in our day-to-day lives without us even knowing about them. For example, bacteria perform fermentation by converting carbohydrates into lactic acid. Yeast perform fermentation to obtain energy by converting sugar into alcohol. And some examples of industrialized products from fermentation are beer, wine, yogurt, cheese, kimchi, biofuel, and even hydrogen gas. Fermentation occurs naturally, so for many years, people would use fermentation to make products such as cheese, wine, bread, and beer without even understanding the biochemistry behind it. It wasn't until the late 1850s that a scientist by the name of Louis Pasteur became the first scientist to actually study fermentation. In his study, he was able to show that fermentation was caused by living cells. However, it wasn't until 1897 when a German chemist named Edward Bochner grounded yeast, extracted fluid from them, and then found that liquid could ferment a sugar solution. So, how exactly does fermentation occur? To understand this, let's dive deep and see what exactly happens at a cellular level. It is no secret that pretty much everything in existence needs oxygen to survive. The cellular respiration formula has an input of glucose and oxygen that leads to the generation of carbon dioxide, water, and ATP. And ATP, as many of us know, has the ability to power many cellular processes. The stages of cellular respiration include glycolysis, pyruvate oxidation, the Krebs cycle, and oxidative phosphorylation. However, what exactly happens when a cell is unable to use oxygen? Well, in that situation, anaerobic respiration occurs. Anaerobic respiration occurs in the absence of oxygen, and there are three situations when it's likely to occur. One situation is when the cell lacks a final electron acceptor to carry out cellular respiration. Another situation is where the cell lacks genes to make appropriate complexes and electron carriers in the electron transport system. The last situation is when the cell lacks genes to make one or more enzymes in the Krebs cycle. Whereas the lack of an appropriate inorganic final electron acceptor is environmentally dependent, the other two conditions are genetically determined. So what exactly does the cell do in this situation? Well, one solution is to adapt and use an alternative compound. An example of an alternative compound that bacterial cells use is sulfate. This process is called sulfate reduction, and it utilizes sulfate as a terminal electron acceptor in the electron transport chain. The second solution, and the one that we're primarily going to focus on, is fermentation. In this process, the cell focuses on glycolysis to produce ATP by reoxidizing NADH2 NAD+ to be used as an electron carrier. Fermentation does not involve the electron transport chain and does not directly produce any additional ATP beyond what is already produced during glycolysis. This process produces a maximum of 2 ATP molecules per glucose during glycolysis. Now let's look at the different types of fermentation. The first type of fermentation that we're going to be looking at is called alcohol fermentation. In this, yeast begin with sugars such as glucose, fructose, or sucrose, and finish with carbon dioxide and ethanol, while producing small amounts of ATP. The primary purpose of alcohol fermentation is to produce ATP under anaerobic conditions. The whole process can be broken down into two steps. The first process includes a carboxyl group being taken away from the pyruvate and released as carbon dioxide, which produces a two-carbon molecule known as acetaldehyde. In the second reaction, catalyzed by the enzyme alcohol dehydrogenase, transfers an electron from NADH to acetaldehyde, producing ethanol and NAD+. Ethanol fermentation is used for the production of alcoholic beverages, for making bread products rise, and even for biofuel production. The second type of fermentation is called lactic acid fermentation. It occurs when pyruvate molecules from glycolysis are fermented into a substance called lactic acid. Lactic acid is an organic acid that is found in both animal and bacterial cells. In animals, it occurs in muscles when the tissue requires energy at a faster rate than oxygen can be supplied. You can think lactic acid when you feel sore and tired after your workout. In bacterial cells, lactic acid fermentation is used to convert lactose into lactic acid in yogurt production. During yogurt and cheese production, the highly acidic environment generated by lactic acid fermentation denatures milk proteins, which causes it to solidify. When lactic acid is the only fermentation product, the process is said to be homolactic fermentation. However, many bacteria perform heterolactic fermentation as well, in which they produce a mixture of lactic acid, ethanol, acetic acid, and carbon dioxide as a result. It is important to recognize that in addition to lactic acid fermentation and alcohol fermentation, countless other fermentation processes occur in prokaryotes, with the primary purpose of maintaining a proper supply of NAD+ for glycolysis. Without this mechanism, glycolysis would not occur and no ATP would be produced from the breakdown of glucose. It is also important to understand that most forms of fermentation, besides homolactic fermentation, produce gas, commonly carbon dioxide and or hydrogen gas. Varying types of fermentation methods are also used in food production, and each results in the creation of different organic acids, which contributes to distinct flavors of specific fermented food products. Okay, so now let's look at some real-world examples of fermentation. So quite often in the lab we use acetone and butanol. Well, this is produced during a acetone-butanol-ethanol fermentation process. When it comes to pharmaceutical compounds, such as antibiotics, vaccinations, and vitamins, these things are produced through a mixed acid fermentation. We can even distinguish between various types of bacteria and microbes based on their ability to ferment certain substances. So that basically sums up a fermentation. Now let's go ahead and look at some review questions to make sure that we kind of got everything. Feel free to pause the video, go through these questions, write them down, and then there will be an answer key at the end of the presentation.